Alright, sit back, get comfortable, and let's make coat colors complicated with coat color genetics. Alright, starting with W and G. W, white, and G, gray, represent alleles whose actions can obscure the actions of other coat color genes. If either the W allele or the W gene or the G allele of the G gene are present, then the coat color genes cannot be determined by superficial examination. Dominant W. These horses typically lack pigment in its skin and hair at birth. Skin pink, eyes brown or blue, and the hair white. Such a horse is termed white or albino. Non-white horses are WW, or little w, little w. Foals born big W, die shortly after birth. More on that later. Dominant G. Born any color but gray, and will gradually become white or white with red or black flecks, known as flea bites. Evidence of graying may appear around the eyes and nose of the foal. In contrast to big W horses, gray horses have pigmented skin and go through lightening stages. Gray horses will be either dominant G or big G or big G, little g. It's not possible to tell if a horse is homozygous for G. All non-gray horses will be little g, little g. For homozygous recessive colors, both alleles are written in the notation for color assignments. Since a horse showing a color or pattern produced by recessives is by definition homozygous for the recessive alleles. Because gray is produced by a dominant gene, at least one parent of a gray horse must also be gray. If a gray horse does not have a gray parent, then the purported parentage should be considered incorrect and seriously reevaluated. The E gene. The allele of the E gene contains the instructions for placing black pigment in hair. These hairs may be found in a distinctive pattern on the points, such as the legs, mane, tail, and tips of the ears, or over the entire body aside from white markings on the face or legs. The alternative allele to big E is little e. The little e allele allows black pigment in the skin, but not in the hair. The pigment conditioned by the little e allele makes the hair appear red. If there is no black pigmented hair, then the horse has the genetic formula little e, little e. These horses will range from dark liver to lighter chestnut or sorrel, and all shades in between. Manes and tails may be lighter, flaxen, darker but not black, or the same color as the body. If a horse is not gray or white, its genetic formula is little w, little w, little g, little g, little e, little e. If two red horses are red to one another, the resulting offspring should also be red. If the offspring has black pigment, big E, or is gray, big G, or white, big W, then the assumed parentage is incorrect. The A gene. The A gene controls the distribution pattern of black hair. The A, in combination with E, will confine the black hair to the points to produce a bay of any variety of shades. Any bay horse will include big or A and E, as well as little w, and little g, little g. The alternative little a allele does not restrict the distribution of black hair. Thus, in the presence of the big E allele of the big E gene, a uniformly black horse is produced. In most breeds, the A allele is very rare, and thus black horses are infrequently seen. Neither big A nor little a affect the pigment or the distribution of black hairs. So, an examination of coat color cannot determine which alleles of the A gene a red horse has. The C gene. An allele of the C gene causes pigment dilution. Fully pigmented horses are big C, big C. Heterozygous horses, big C, big C, have red pigment diluted to yellow, but black pigment is not affected. A bay, big E, big A, will become a buckskin by dilution of the red color body to yellow without affecting the black color of the mane and tail. The genetic formula for a buckskin, which is what this is little w, little w, little g, big A, big E, big C, big C. A red horse, little e, becomes a palomino by dilution of the red pigment in the body to yellow, with the mane and tail further diluting to flaxen. The genetic formula for a palomino is little w, little g, little e, big C, big C. A genetically black horse, big E, little a, can carry the dilution allele without expressing it because big C only affects red pigment. Homozygous big C, or CCR, completely dilutes any coat color to a very pale cream with pink skin and blue eyes. Such horses are often called cremello or perlino. Typically, such horses are the product of mating two dilute-colored animals such as a palomino or a buckskin. The D gene. Not to be confused with C, the D gene determines the second kind of dilution. D dilutes both black and red pigment on the body, but does not dilute either pigment in the points. Red body color is diluted to a pinkish red, yellowish red, or yellow. Black body color is diluted to a mouse gray. In addition to pigment dilution, a predominant characteristic of the D allele is the presence of the primordial markings, dorsal stripe, transverse mark over withers, zebra striping on legs, and cobwebbing on the forehead, also known as dun. D for dun. In an otherwise red horse, the D allele produces a pinkish red horse with darkened red points known as a red dun. Little w, little g, little e, big C, D. In an otherwise bay animal, the D allele produces a yellow or yellow-red animal with black points known simply as a dun. Little w, little g, big E, big A, big C, big D. In an otherwise black animal with the D dilution allele is a mouse gray color with black points known as grula, or if you rather, gruya. Alright, little g, big E, little a, big C, big D. D is found only in a few breeds of horses, and probably in the U.S. only seen in stock horses and some ponies. Other important genes that you should know and be familiar with are N for roan, S for silver dapple in Shetland ponies, and Rocky Mountain saddle horses too. T and O for Tobiano and Overo, respectively. Okay, so in summary and maybe plainer English, W as in white, dominant W is lethal, or double dominant W is lethal. Big W, little w, horses is typically pigmented in skin, hair, and eyes, and appears to be white. Little w, horse is fully pigmented. G as in gray. Big G, horse shows progressive silvering with age to white or flea-bitten, but is born any non-gray color. Pigment is always present in skin and eyes at all stages of silvering. Big G, little g is the same as big G. Little g, horse does not show progressive silvering with age. E, as in ebony. Big E, horse has ability to form black pigment in the skin and hair. Black pigment in hair may be either, pardon the typo, in a points pattern or distributed overall. Big E, little e is the same as EE. Little e, horse has black pigment in skin, but hair pigment appears red. A, as in Auburn. Big A. If a horse has black hair, big E, then that hair is in a points pattern. Gene A has no effect on red double E pigment. Big A, little a is the same as double A. Little a, if a horse has black hair, big E, then that black hair is uniformly distributed over body and joints. Gene A, big A, has no effect on red, little e pigment. Ooh, no. There we go. C as in cream. C, big C, horse is fully pigmented. Big C, CCR, red pigment is diluted to yellow. Black pigment is unaffected. CCR, both red and black pigments are diluted to pale cream. Skin and eye color are also diluted. D as in done. Big D, horse shows a diluted body color to a pinkish red, yellow or mouse gray color, and has dark points including dorsal stripe, shoulder stripes, and leg barring. Big D, little d is same as big D, and little d, horse has undiluted coat color. Alright, so, again, very quickly, in the last minute or so. W, G, bay horse, black horse, chestnut horse, buckskin horse, palomino, cremello or perlino, dun, grula, red dun. And so there we are, coat color genetics in very brief terms.